Koto, tēnā koto katoa. O Vivian Handa Ho, no mai, haere mai, ki te Aotearoa Food Safety Science and Research Centre. Ka nui te mihi nui kia koto katoa. My name is Vivian Hunt, and on behalf of the New Zealand Food Safety Science and Research Centre, I would like to welcome you all here today to our Spotlight on Food Safety Science seminar. Today we're delighted that Graham Fletcher has agreed at rather short notice to give a talk on front-footing food safety using modern research techniques. As many of you all know, Graham leads the Food Safety and Preservation Research team within the New Zealand Institute for Plant and Food Research. His team supports New Zealand industry by carrying out research on the microbial safety, post-harvest processing and preservation of seafood, and on the safety of fresh horticultural produce. He also supports international development programs, ensuring that vegetable producers have the appropriate tools to ensure the food safety of their produce. Graham has a wealth of food safety research experience, which started in 1980, and he's the author of over 70 peer-reviewed scientific publications. I just heard he's got another one out. And he's been a regular speaker at New Zealand and international conferences. So he's an expert in the field, and he's going to be talking about a topic that's obviously front of mind right now. So Graham, over to you.
Thanks, Vivian. So tēnā koutou katoa, ko Ruapekepeke te manga, ko Waiomio te awa, ko Tautiranga te iwi, no Kawakawa aho, ko Fletcher toku whanu, ko Graham toku ingoa. Tēnā koutou, tēnā koutou, tēnā koutou katoa. So I'm going to be speaking about food safety and trying to put it on a front foot rather than on a back foot. Typically, we tend to think about food safety, and in industry, they spend a lot of money making food safe, but they don't advertise it. It's not a part of their marketing brand because everyone takes it as a given. But when you look around the world, you find that food safety is not a given, that we regularly have outbreaks of illness from food. And so this potentially should be a marketing advantage. And what I want to think about today is, can we turn food safety from something that we hide in the back cupboard as something that everyone has to have, but doesn't necessarily provide us any financial benefit, to a marketing advantage. And thinking about that, I think that there's three things come to mind that could be of benefit. One is a trusted partnership. In New Zealand, we have quite a strong relationship between our regulator, MPI, industry, and science. We have regulators and industry that want to take a science-based approach to food safety. Another thing that's really important to put food safety on the front is taking a risk approach, and both our industry and the regulator take risk approach to food safety. And that's really important. I think probably, so science has a lot to do with risk assessment and identifying prevention methods. Then we've got risk communication, understanding what food safety risk can be communicated to the consumer, to the markets, to the retailers, to the wholesalers. And I think probably COVID has probably helped us in this regard. So in New Zealand, we have learned that elimination does not mean zero. And that's something that's important to understand with risk, is that there is no such thing as zero risk, but we want to be able to market ourselves as having an acceptable level of risk, and to be able to communicate that. The last thing about getting food safety up in the front foot, I think, is needing to have an integrated whole supply chain systems approach. We all think about, know about from farm to fork, but in the industries where I work on, it's much more about orchard right through to the fruit bowl. From what happens on the fields, what's happening in the soil, interactions between the soil and the orchards and neighboring properties, through transport, through harvesting, through packaging, through shipping, right through to the consumer. And that we have to have a systems approach right through that system. And for seafood, it's taking it from our sustainable seas right through to satisfying servings and having customers that are satisfied with food that is safe, and that they're proud to know that it's safe. What I'm going to do today is look at 3 case studies, the first two of which are cases where we really have minimal risk of foodborne illness. listeria on apples. So there's been one outbreak in the history of the world that's been attributed to apples, and then listeria on mussels, and again, one outbreak in the world, but these are huge market access issues for the country. And then the third one is on Vibrio and shellfish, and here, internationally, this is a relatively more common situation of causing illness, whereas in New Zealand, it's very much an emerging situation. And I'll talk about those 3 case studies. So let's start with listeria on apples. So thinking about research, in October 2014, Pipfruit New Zealand, which is now New Zealand and Apples and Pears, started an Apple Futures 2 research program. This was an ambitious 7-year research partnership program, which is shortly to finish next year, and it was to support market access. And perhaps almost controversially, they chose to put food safety in their market access program. Controversially because apples are not associated with food safety risks. So they started that program in October 2014, and then in October later that year, in late 2014, 2015, there was an outbreak of listeriosis from apples in the US. There were 35 illnesses, 7 deaths, 1 miscarriage, and they found that there was a common food relating to this, which was caramelized apples. These are and from sort of an English background, we have toffee apples, but in America, they go one step further and they put caramel, sticky caramel and coatings and have these things at festival times. The apples were produced in 3 different pack houses.
Amor, eu preciso ver isso aqui.
And whole genome sequencing confirmed that the whole apples from Biddart Brothers were the original source of the contamination. So although it was research suggested that what actually caused the problem was interaction between the stick and the going into the core of the apple, and the interface between the caramel and the apple that allowed the listeria to grow, but it was blamed on the apple, the original apple, as the cause of the outbreak. Had huge impact on industry. So at that stage, the US Apple Association suddenly called food safety their top issue. And there were several lawsuits filed against the Biddart Brothers. I haven't seen any outcome of that, but presumably court. So what is listeria? It's a bacteria. It's found everywhere. It's not like a lot of foodborne illnesses come from bacteria that only grow in mammalian or animal, in warm-blooded animals, but this is found everywhere. It grows in moist environments, including soil and decaying vegetation. It's very hardy, salt and acid tolerant, can survive and grow at low temperatures, so it can grow in refrigerated temperatures. And it also grows and persists quite well in food manufacturing environments. So often the manufacturing environment or the packaging, pack house or the processing facility is the source of the contamination. And that was the case in the Biddart Brothers case. It causes listeriosis, which is an infection, and it causing meningitis and miscarriage and death. Only affects a particular risk group, which are people with immune and compromised systems, including pregnant women and their babies, the elderly, and others who, for various reasons, have a weak immune system. Not many cases happen in New Zealand or anywhere, but it's one of the leading causes of death from foodborne illness, and typically 15 to 30% of cases die. So it's pretty serious. In New Zealand, we have actually one of the highest reported incidences in the world, with 0.6 cases per 100,000 people. And there's the number of cases or notifications over a 20-year period. And you see that we average maybe 20, 25 cases a year. Most of the cases are not related to pregnancy and perinatal. Most of them are other immunocompromised people, particularly the elderly, that suffer from listeriosis in New Zealand. What are the regulatory requirements? Some jurisdictions have an absolute standard for Listeria monocytogenes. So the US Food and Drug Administration, for example, They treat Listeria monocytogenes as a contaminant, so they say it must be absent, and that means in practical terms, in every 25-gram sample, it must be absent, which for science is really difficult because we know that we can test any number of 25-gram samples and find it absent, but the next one it might be positive. And it's just not a realistic standard, but that is the standard that our industries have to meet if they're sending to countries like the US. And Australia and New Zealand, we have joint food standard authority, and they take a bit more of a risk-based approach, and they have two, they recognize that listeriosis requires susceptible people to consume a high number of monocytogenes. And so there's two standards, one for ready, or these are criteria rather, and one for ready-to-eat food. And if the Listeria is considered that it can grow in the food, then again, quite similar to the US, it must not be detected in a 25-gram sample. And that's considered potentially hazardous. But in foods which it will not grow, then there's a different standard, and that, or different criteria, and that is that it must have less than 100 organisms per gram, and that's considered satisfactory, unless a listericidal process is in the system which should have killed it, in which case it should have got to undetectable. So, given those two standards, the first question to ask, well, which category do apples fall into? And a PhD student in our lab, Agam Nangal, has been looking at this and looking at Listeria monocytogenes through the supply chain and question, does it grow? One of the questions of his PhD. And he inoculated apples with large numbers of Listeria and subjected them to a typical supply chain, one going to Europe, one going to the USA, and found that in both cases, the Listeria declined. It did not grow. And then he took some of those apples at each of those times and put them up to room temperature, 20 degrees, and again, looked to see and held them for 8 days at each time and said to see, will it grow? No, it either declined or it stayed static. So for apples, unless you dip it in candy and stick a stick into it and do strange thing to it, for ordinary apples, Listeria does not grow. So the less than 100 per gram is a reasonable standard for apples that are going to be consumed as fresh fruit. Over the last 6 years, we're part of our work in the Apple Futures 2 program. We've sampled a wide range of orchards and packhouses, testing for foodborne pathogens and indicating bacteria, including Listeria. And unsurprisingly, we did detect some Listeria on apples before washing. So we can go to a field, we can pick some apples, and sometimes we will find Listeria on them, Listeria monocytogenes, a human pathogen, because it's a soil-borne organism. And then the question is, what's the risk from this? So standard testing evaluates the presence in a 25-gram sample, and you see the standards are written around that. However, unless the fruit is damaged, all the contaminating bacteria are on the surface of the fruit. They're not, at least they're certainly not found in the white flesh. And for our samples, what we did, rather than taking a 25-gram sample, we removed the bacteria from the surface of 3 apples, which is about 900 grams. So we have 900-gram samples, and we typically do triplicates of that. So, so three of 900 grams. So each apple is about 100 grams. And what do we find? When it's present on apples in the field, we find that Listeria monocytogenes typically at concentrations of less than 0.01 per gram, much less than the 100 gram permitted in ready-to-eat foods on which it will not grow. So unless you do something strange like caramelize your apples, this is not a health and safety risk, not a food safety risk. But it's still a market access risk, and that someone could do some tests and find that that Listeria, and we can get recalls, could get recalls. So can we eliminate it to meet zero tolerance? And here we're looking at hurdle technologies. Hurdle technologies are where we combine the sublethal doses of different hurdles to achieve lethal inactivation. And after a lot of preliminary studies, we ended up combining mild heat and sanitizer in the water flumes. When apples are moved around a pack house. They often initially moved in water because this is an easy way that doesn't bruise them. And the water, they sanitize the water to make sure no pathogens are growing in there. And in some cases, some of the water they actually heat a little bit in order to be able to wax the water. And what we looked at increasing the temperature of the water, and it was actually the hot water treatments that we've been evaluating have primarily been done for phytosanitary purposes to get rid of insects and fungi and things, rots. But we're looking at it in terms of food safety, and we looked into to find out how hot can we put apples in water without it cooking the apples or damaging it. And we chose temperatures that would work that and combine it with sanitizers. And what we achieved was quite satisfying. So here we've got an experiment where we apply a drop of highly contaminated drop onto a surface of an apple. And then if we wash it, we get about a 2 log reduction, reducing that. If we put it in the sanitizer, we come down to further. And then when we combine a sanitizer with a hot water treatment, this was at 51 degrees, we get very dramatic reductions. That was when we put a drop, as I said, on the surface of the apple. But if we immerse the apple in contamination, like if the water that is floating has become contaminated, and some water will get into the calyx, etc., and the stem ends, we get less reduction, but still. So here we go from contaminant, washing it, applying the sanitizer, and then sanitizer in hot water. Much more difficult to inactivate, partly because it's protected some of the bacteria protected in the calyx and in the stem ends. And we're currently, this year, we've been carrying out pilot studies in a pack house on this technology. Now I want to think about genomic studies and how these help us gain food safety assurance. And so Listeria monocytogenes whole genome sequencing. And here we've got the New Zealand Food Safety Science and Research Centre has a project whereby as many as we can get our hands on Listeria isolates from New Zealand have been mapped. Their whole genome sequences have been determined. And then and here we've got a map of the whole genome sequences, the MLST, multi-locus sequence typings at this stage we had 800 different strains of Listeria from New Zealand sources, where it's seafood, horticulture, clinical, meat, poultry, and others. And here they all are around. And you see that the red ones there, these are most of the clinical outbreaks. And there are 3 main lineages of Listeria monocytogenes. There's lineage 1. And most of the outbreaks, or most of the, not outbreaks, but illnesses, clinical cases are coming from in that lineage 1. And then there's lineage 2. And most of the environmental strains come in lineage 2. And a lot of our seafood strains that we get were in lineage 2. Some clinical isolates still happen in lineage 2. And then we have here lineage 3. And there's also lineage 4, which we haven't detected. It's quite rare. And if we look at lineage 3 a bit more, that's quite we're quite interested because we found quite a lot of our horticultural isolates were in lineage 3, which was a little bit unexpected at the time. It's very rare, it's rarely associated with clinical cases. And it was thought to, when we started work, it was thought to be a rare lineage, but a recent study in the US found it was actually the most prevalent lineage in US soils, lineage 3. So that was interesting. Within that lineage, we noticed that a lot of our samples were of a particular sequence type, all very similar, closely related. Sequence type 299. There's some statistics about it, no plasmids on it. But when we studied that, looked at that sequence, when you look at that sequence type, we find that it contained a stress survival islet. So that enabled it to grow and survive under very suboptimal conditions, which might be why it's dominating in some soils, etc. There were no markers for heavy metal or detergent tolerance or antibiotic sanitizer tolerance. And when we looked at the virulence markers within the sequence type, there were huge absences of the 93 virulence markers that are associated with causing illness with humans. Only 52 of them were present in this sequence type, and some of the important ones that enable a Listeria monocytogenes to infect a human were absent, so it lacked capability to infect. So although it was common in our horticultural, in our apples, of a few times we detected it, it wasn't going to infect humans. But we still want to get absence. So where did they come from? Those horticultural produce of sequence type 299, 12 were from, 4 were from fruit. So we had 12 isolates, 4 were from fruit, 4 were from wash water, and 4 were from equipment. For apples, all apples going through a New Zealand pack house, they get part of the process is they get a high pressure wash to remove a whole lot of stuff from them. And when we carried out core genome multi-locus sequence typing, we find that here the key here, we got our 4, these are our 12 isolates, how they relate to each other, whether it's region A or region, we had got them from 2 regions, and region, and some were on the fruit, and here we've got some on the fruit, and the same genotype was on the equipment. But if we look at the ones that were on the water, they were quite different genotypes. So this suggests possible cross-contamination from contact surfaces rather than the water, or it may be that the fruit, quite possibly the other way around, the fruit brought it in, and it's contaminated the equipment, the surfaces. So we can learn a lot from whole genome sequencing and tracking down where different Listeria come from and what it means and what the likely sources are. On the other side, the regulators can learn a lot, and the epidemiologists can learn a lot about where an outbreak has come from and what was the causative product. So let's switch now and think about Listeria in mussels. So as I said, there was that one situation in apples where we've had outbreaks from candied apples. And mussels, Listeria monocytogenes is not a marine organism. And again, mussels have only been implicated in 1 outbreak of illness in the world that I've been able to track down. And unfortunately, it was New Zealand. It was in 1991, it was smoked mussels. And when it was investigated, it found that the contamination came from a particularly poorly managed processing facility way back in 1991. However, because of zero tolerance, subsequently we've occasionally had, I think on one occasion at least, had a recall where the US picked up Listeria monocytogenes in our mussels arriving in the US. Even though the product is labeled to be cooked and such things, they'd said, no, we're not accepting it, and got sent back a whole shipment of mussels. But currently it's very rare in mussels, but the occasional presence of Listeria in New Zealand seafood has led to huge compliance costs. And back in the day after that one recall, just one company had to carry out 20,000 environmental swabs and more than 2,500 product tests, a huge sampling program. And even today, if we talk to the industry, they spend hundreds of thousands, or maybe $1 million a year. Companies spend on testing to make sure that their mussels or seafood does not have Listeria in it. And in that case, with all that testing program, there were only about maybe 70 positive samples among the 20,000 tests. And again, always whenever we do the tests, we find out that the concentration is at really low levels, with typically less than 10 per gram, compared to a standard of an infectious standard of 100 per gram. But Listeria will grow on mussels if it's, but not usually on the frozen mussels that we export. At that time, we looked at the different strains. We were using an older genomic technology then, pulse field gel electrophoresis. We found, of those strains, we found 27 different pulsar types. but only three of those persisted in mussel factories. These three persisted for more than 6 months. So they were quite, and one of the problems is that, as I said earlier, Listeria monocytogenes will grow in a processing facility, and that's a major source of contamination. And these ones, try as they could, it took a long time to get these to get on top of the Listeria. But when we looked at the infectivity of those three different pulsotypes, they were very different. This is the ability of these pulsotypes to invade human gut cells. And this one here had very low invasive ability. And this pulsotype, which is mostly, when we do the whole genome sequences, it's mostly sequence type 321, we found out that it carried an internalin A truncation. Internalin is the gene that codes for an enzyme that enables the Listeria to penetrate and get into the human gut cell. And this stop codon was observed in this pulsotype. So although it was persisting in factories, there was very little chance that it would cause illness. And then subsequently found that 32 of 139 isolates also lacked another of the internalins that's needed for invading human cells. So when we look at again at the genome's diversity found in New Zealand, here's the sequence type 321 and these are all the seafood isolates that we got of them. Okay, so how have we managed to control? That's a pity. I think I've lost. I know what I've done. Sorry. So that sequence type. I'm getting lost. Nope. Okay. Apologies. One of the things that industry have done to clean up their facility is that they've implemented strict temperature control points during the heat shucking of mussels. But some of the biggest gains have come by changing factory design. So in the old days when they were having those problems, a lot of factories were made out of this polyboard with insulation behind it, lots of crevices for bacteria to hide in, and then they've had to rebuild their factory to solid, clean line things where there's very little places for bacteria to hide in, changing the fittings. This is an old factory with the polyboard there, and also used to be all sorts of stuff in a fish processing factory, and places light fittings like this to much cleaner fittings that prevent harborage spaces for Listeria, because Listeria will grow in all sorts of nooks and crannies in the factory. And so that's how they've been able to get on top of, main way of getting on top of that. Just talk about one more sequence type for Listeria, and this is sequence type 204. And this is also was found in seafood factories. And when we look at that one, whereas this one here was had very little ability to invade human cells or but it did persist in factories. This one was quite different. It's lineage 2, and it appeared to be highly persistent and dominate. And in one particular seafood factory, we've mostly found it only in one seafood processing facility. It was first isolated in 2013, and it's been occurred again until 2019. It was found again, and it was the only sequence. So this is environmental sampling that they do, swabbing whole factory to make sure that there's no chance of it contaminating the product. And when they find it, they track it down and get rid of it. The only sequence type, this was the only sequence type isolated in this one facility over that whole period of 6 years. And when we looked at the genomics of it, we found that it contained, again, a stress survival islet, which enabled it to survive under suboptimal conditions. It contained not many virulence markers, but we need to do more work on that one. But it also contained specific markers for heavy metal and sanitizer resistance, and these were on a plasmid, and it contained benzalkonium chloride is quaternary compound, and it was tolerant to that, and it was also tolerant to cadmium, and it had a whole lot of resistance factors that enabled this particular strain to be so persistent and so difficult to get rid of. Lastly, I'm going to briefly talk about Vibrio and shellfish. And this time, as I said, this is more of an emerging issue for New Zealand. So particularly Vibrio parahemolyticus, gastroenteritis from shellfish. So that's vomiting, diarrhea. So it does, unlike Listeria, it doesn't usually kill people. It's usually causes a pretty unpleasant illness for a day or two. But it's Vibrio parahemolyticus is a natural part of the marine ecosystem. It proliferates in warm seawater, and it bioaccumulates in shellfish. So shellfish they feed by filtering seawater and concentrating anything that's in the seawater. And when Vibrio parahemolyticus is there, it can be concentrated. And when shellfish containing high numbers of parahemolyticus are consumed, either raw or lightly cooked, it can cause gastroenteritis. So if we look at, this is US outbreak data here, maybe 3% of outbreaks of the whole country were caused by Vibrio parahemolyticus. So this was in 2017. They had 17 outbreaks from parahemolyticus and about 70 case illnesses. Whereas in New Zealand, we have, up until recently, we've never had well-documented outbreaks of parahemolyticus until 2019. And we had an outbreak in 2019 in June, and we had a second outbreak in 2020, also in June. And in both cases, the implicated product was raw or lightly cooked mussels. People who bought mussels and either ate them raw or didn't cook them properly. Well, the regulatory requirements for parahemolyticus are some jurisdictions, again, have an absolute standard, not absence, but in Canada, for example, they have a standard that it must have less than 100 organisms per gram in live oysters. This FSSRC does not have a numerical criteria, but they do state that the infectious dose, they say, is greater than 10 to the 5 viable pathogenic cells. And we're probably going to have to revise that. So they require seafood to be quickly chilled to maintain them under refrigeration because the organism won't grow below about 10 degrees. And shellfish harvesting practices may have to be implemented to assure that shellfish is not harvested when the water temperatures are in a particular range. And the FSSRC also states that cooking to an internal temperature of 65 will destroy any parahemolyticus. So cooking mussels, got to cook it to reach an internal temperature of 65, not just steam it till it opens, if you want to control that. This is some work that we did looking at the frequency of parahemolyticus across a range of New Zealand shellfish across 7 different harbours over a 3-year period. And we find that these are green lines of the parahemolyticus, we always get a summer peak around February, March of parahemolyticus in shellfish. And it's commonly there, and it correlates somewhat to the water temperatures. Most strains of parahemolyticus are not pathogenic and they don't cause outbreaks. There are a couple of pathogenic pathogenic markers that we look for, and here the numbers of them are much lower. So we're getting low counts, less than 10 per gram of these pathogenic strains within our New Zealand mussels and oysters that we were sampling at that time. However, interestingly, in June, when we're having these outbreaks, now the numbers of parahemolyticus in New Zealand shellfish are invariably low, less than 1 per gram or 10, certainly less than 10 per gram June. So that's so the outbreaks that we've been getting have sort of thrown a challenge to this. We found that these are 6 different harbours, and the relationship between parahemolyticus numbers, there is a relationship to temperature, and sure enough, as the temperature increases, the numbers increase, but there's a lot of spread, and also different harbours were different. So you'll see the shape of these lines are quite different. And if we look back to some work that we did in the 1980s, early 80s, we were detecting much lower numbers of parahemolyticus for the same temperature ranges at that time, although the methods have changed. So when we came to genomic analysis, and I'm grateful to Andy Powell, who's on the line here too today, whole genome sequence showed that most New Zealand Vibrio parahemolyticus strains, 65% of the ones that we've analysed so far, belong to novel sequence types that have not been found in other parts of the world. So we've got our whole unique to New Zealand system. Most strains that cause the outbreaks overseas contain one of two pathogenicity markers, TDH or TRH. Most occasionally there are outbreaks that from strains that don't have these genes, but most of them do. These genes were relative, as we saw in that plot with the green lines, relatively rare among New Zealand isolates, less than 2%, and until 2019 we had not caused notable outbreaks. But the 2019 and 20 both contained both of these pathogenicity genes, TDH and TRH, and they may represent a more virulent strain. The fact that we are having low numbers and causing this illness. And the outbreak strain in 2019 was actually a pandemic strain. We all know about pandemics now. Well, Vibrio parahemolyticus has pandemics. They're not so widely broadcast as COVID, but because they don't kill people. And but it has spread. This strain has spread around the world, and 2019 was the first time it was found in New Zealand. And we're currently looking at wanting to identify the genetic and phenotypic factors that differentiate outbreak strains from environmental. What can we do if we've got parahemolyticus and we want to control it? One of the simple controls is freezing and frozen storage. And colleagues in Oregon State University carried out this trial here and found that if they froze their in this case oysters, and held them in frozen storage, they got log reductions. And they were, for the US, they were targeting a 3.5 log reduction, and they could conclude that if they stored it for 5 months at -21, -18 is a little bit dies a little bit more rapidly, that they can achieve what they needed to achieve. And we've been evaluating freezing as a control for another pathogenic Vibrio species, Vibrio vulnificus, but the naturally contaminated oysters, we found that our results are similar to this. So this can be applied. So control is available if it's needed, but 5 months of frozen storage is a long time for oysters. Doesn't necessarily make them the best product, but there are controls available. I just wanted to say too, I should have mentioned before, when we talked about this getting low numbers, this kind of outbreak, the other possibility is that people have left their mussels warm for a long time and allowed their numbers to grow to above what we find in the seawater. So just by conclusion, I think New Zealand can be proud of our science-based food safety systems. Our research is contributing to both the safety of New Zealand's seafood and horticulture industries and to ensuring and maintaining market access for our product. And sometimes these two are quite different. So safety might be ensuring that our apples have got less than 100 per gram. Market access to some markets means ensuring that it's absent. And the question that I want to know is, can we move towards actively promoting trust in our food safety systems? As a marketing, can we market that? Can we put food safety as a front foot, something that we sell New Zealand food on, that we're not only sustainable, we're COVID-free, we hope, and soon, but also we're safe. Can we get away from having food safety in the back cupboard that we have to spend a lot of money on but can't market? Can we put it up the front and say New Zealand food is trustworthy because we have such good systems? Just need to acknowledge a whole lot of people for a lot of this work. The seafood and horticultural industries supporting the work, colleagues at Plant and Food Research collaborators for the epidemiology, Jackie Wright and co. for whole genome sequences, with the ESR, did a lot of our whole genome sequences at ESR, and also the University of Surrey, Arnold van Vliet, who I see is online too, did some sequences and has helped with the analysis of the genomics. The CFIS in the UK, and Andy Powell, I said he's online. And also, so thank you for our collaborators, and also funding the New Zealand Food Safety Science and Research Centre, who's been doing that whole genome sequence program. MB, who funds the Cawthron Safe New Zealand Seafood Program that we do a lot of our Vibrio work under and our Listeria seafood work under. MPI, who's been funding us to do looking at sequence type 36 monitoring this year. And then the Australian Research Council, who funded Agam's PhD thesis, and Mark Bradbury, my co-author, who's a postdoc in our laboratories from the Australian Research Council. So, thank you.
Thank you very much. That was a great talk. And if everyone's got questions, please put them into the chat. I have one from Kim, who got off early on the discussion about the apple. He wants to know, does the hot water affect the apple eating quality of the water?
No, it doesn't, and we've done our plant physiologists and things have done a lot of work. Post-harvest people have done a lot of work to make sure that we don't heat it up to the point that it does affect it, either the eating quality or the storage quality. If we heat it too hot, yes, it does, and we have storage diseases and things, but at the temperature that we're looking at, no, it doesn't.
Okay. Another question from Kath is, what other control options are available to industry to control Vibrios? High pressure processing, marination? To control Vibrios, yes, there are other options. So high pressure processing is one. There's a hot water treatment that is approved, but it does produce would be okay for mussels, but for oysters, it produces a slightly cooked product, which you probably don't want. Radiation is always a possibility. They're Vibrios are actually very sensitive, and that's why we can use frozen storage. Most food poisoning bacteria, you can't use frozen storage to control them, or they won't control them very much. But for Vibrios, they're quite sensitive and relatively easy to kill. Okay. Question from Arno is, Graham, can we say the food is really safe? I'd like to have it safe.
No, we can't say that a food is safe. That's why we need to take a risk-based approach. We can market as being the safest food in the world, maybe, but we need to a product that we can trust the food safety of it, because there is no such thing as absolute safety, and that just has to be. And we need to, with consumer education and things, we need for the world to understand that there is no such thing as absolute safety or absolute anything.
Yeah, no, it's a good point. I think, especially in the current situation, and New Zealand does have a good reputation. But one concern I had was, why do we have the highest rate of highest reported incidence of listeriosis?
I'm not sure on that. I'm not sure if anyone else online knows that. It may be that we've got good reporting and capturing systems, but there's probably more than that too. I'm not sure. We unfortunately top the world in a couple of other things. Campylobacter is our big one, where we're really great at Campylobacter and making people sick with Campylobacter, although we've done a lot of work to improve that. Whereas others where people have a lot more problem with, like Salmonella, it's much less of a problem in New Zealand compared to other countries. And as I said, Vibrios around the world have been a significant problem, but until recently have not been a problem in New Zealand. So yeah, I'm not sure.
Well, a question from Tim is, why do you think the virulent Vibrio parahemolyticus strain is causing a problem in winter? Yes, because I noticed it's in June both times. It seems a bit strange.
Exactly. We're not sure about that. They've got a current program with NPI trying to understand the distribution of this strain, and we carried out a sampling program over this from January to June this year, and we actually didn't find any of it until we didn't find any of it, but we did find the strain that caused the outbreak this year, but it was at very low numbers. But one suggestion that I had from Jackie Wright, I don't know if she's online, is that come winter, a lot of people take like mussels because of their ability to deal with arthritis, and so comes winter, you get arthritis coming in, so people might be eating more raw mussels because it does help with arthritis, but that's just a guess. So, but we don't know.
Another question from Kath is, how much reliance can one place on whole genome sequencing and sequence data on the virulence, antimicrobial resistance in terms of predicting how the bacteria might behave in reality?
I think it's part of the story. So you might have noticed when we did the virulence, we did do whole genome sequencing, but we also did invasion assays of human gut cells. So it's only part of the story. It shows potential, but you've got to look at epidemiological data, and so some of those strains, we have seen them in clinical isolates. It's difficult to know whether that was the actual causative organism in the clinical isolates. That some of those strains that are certainly less virulent.
Okay. And Thomas Salisbury says, are there moves being taken within the regulatory bodies to make microbial limits target instead of a generalized Listeria monocytogenes for the gen types which impact virulence also be included in the future?
I don't think so. I don't think there are regulatory moves. We would advocate thinking about that, but there are cases where we do that. So, for example, E. coli. E. coli is a natural resident of the human gut, and there there are regulations for E. coli because it indicates potential contamination, hygiene damage problems, but the limits are quite high. You can have 100 per gram or 1,000 per gram even for some products of E. coli. But where we have virulent strains of E. coli, such as E. coli 0157, then it has to be absent. So there's a case, in those cases, we do differentiate between virulent and avirulent strains of, in that case, E. coli. So there is a precedent there. In Listeria, virtually, by far the dominant number of outbreaks are caused by Listeria monocytogenes, whereas other Listeria species, people test for them, and some people take action based on Listeria monocytogenes species, but they're not regulated for typically.
Okay. Someone has just asked a question, Kathy, could the Vibrio come on ships, ballast water from warmer areas overseas?
It could have done, yes. That's always a consideration. So, I won't tell where this outbreak came from, but it was near, not too far from big ports.
But interesting that the Vibrio that's been that you've done speaking work on here is completely different.
Yes, a lot of the strains were completely different, which I think was probably a surprise. I'm not sure. Andy did that whole genome sequencing, and I think that was a surprise to find how many were different. You expect a few different every now and again, but the bulk of them were different. And yeah, so we are a bit of an island, and that had made a big impact on our microflora.
Exactly. Okay, well, look, I think people are, any further questions from anyone? If not, I'll close this. We haven't got a speaker sorted out for the next seminar, which is going to be on the 25th of September. So if any of the scientists on the call would like to send an email to me and suggest a topic, that would be great. And otherwise, I might shoulder tap somebody. Just watch out to see an email from me. And yes, so thank you very much, everyone. And thank you especially to Graham and your co-authors, Mark and
Shivani. Yep.
Shivani. So thank you very much for a really interesting talk. That was great and fantastic to see the work that's been done to understand how whole genome sequencing can inform control measures as well. So thank you very much, everyone, for calling in. And I think we got a record today. We had over 80 people on, 82. So thank you.
Oh, great. Thank you all for joining us.
Thanks. Cheers, everyone. Bye.
Bye.